Hello, this is Mr. Buss, and I'm going to go over the activity we did in class, the 6B RNA beads activity. So in this, we had quite a few materials. This is one of them. This is just a paper copy, but it's of a DNA strand, right? So what we're going to do in this activity is we're going to walk through the process of transcription and translation. And so for transcription, right, we need a DNA strand. So we're going to go to the nucleus, and in the nucleus, we're going to find a double-stranded DNA helix, and one side of that DNA strand is going to be the template for RNA, or specifically messenger RNA, right? So that template strand is given to you. So the top strand here in the 3 to 5 direction is the template strand, which will be red in the 3 to 5 direction, and the mRNA that's produced is going to be produced in the 5 to 3 direction. Now, I don't have a cutout for RNA polymerase. That's the enzyme that would do this, right? So the top strand is the template strand, and of course DNA has thymine, adenine, cytosine, and guanine. But the RNA strand that we're going to make is going to have uracil, adenine, cytosine, and guanine, right? So I'm just going to use the same color, whether it's thymine or uracil. And we're going to still use the same idea that the pop beads have directionality to them, and we're going to use this 5' to 3' directionality. So if I'm going to form this bottom strand, I'm going to make sure that the sugar phosphate backbone, all of the pop beads are going to be pointing in this direction, in the 5 to 3 direction. And so I've already made that for you, so you don't have to pause the video while I make that or anything, or fast forward the video. So here is the mRNA that would have been made from this DNA template, right? So mostly fits on there pretty good. Colors are a little off just because the pop beads are just a little bigger than the template. So that mRNA is made in the nucleus. That's transcription. RNA is single-stranded, right? And it has uracil instead of thymine, and ribose sugar instead of deoxyribose sugar. Also, it's much shorter, right? So DNA is very, very long continuous. RNA is just part of the DNA that codes for a gene, or a gene that codes for a trait. So there's our mRNA. Transcription is over. And next, what we're going to do is we're going to do translation, and that happens at the ribosome. So we have a ribosome cutout that we're going to use for this activity, right? The ribosome is going to hold everything together. It's going to hold the mRNA, and it's going to have spots for the tRNA cards that we are going to have to go in as well. So before we actually do this step, what we're going to do is we're going to charge the tRNA molecules. So here's our tRNA molecules, and they are all lacking amino acids, okay? So tRNA stands for transfer RNA, so there's a little T out in front of the RNA there, and they're made of RNA, and each of them has a 3 base pair anticodon, right? So these are the anticodons, and in class we've learned how to use the code wheel or the code chart to read mRNA. So this is the code chart for mRNA, but these are the anticodons on the tRNA. So what we have to do is we actually have to kind of like reverse engineer this, right? So we're given, like for this one here, GCC is the anticodon for this one right here in the bottom left. Well, the codon would be CGG, right? Cars park in the garage, C to G. So I'm going to look up CGG on the code wheel, CGG, and that's arginine. CGG is arginine. No, look at that. I've got an arginine amino acid, so that's charged now. That takes a little ATP for that to happen. This is going to happen in our cells, in our cytoplasm. This one in the bottom middle is UCA. UCA is serine. Look at that, I got a serine card. This in the bottom right is ACU for the tRNA, so mRNA would be UGA. UGA, stop. Now I'll just do these quick. CCG would be GGC. GGC is glycine. UAC. UAC. Wait, I almost did it wrong. That's the anticodon, UAC. Look up the codon, AUG. A-U-G, methionine. And I've only got one more amino acid left. That would make this proline. Okay, so I've charged my tRNAs with amino acids. So those are ready to go. I've figured out the right one. Now there's, in our body, we have 64 different tRNAs because there's 64 different anticodons to the 64 different codons. There's only 20 different amino acids, and so each tRNA holds a specific amino acid. This tRNA in the bottom middle will always hold serine. Right? And it's just going to be recycled, reused. Sometimes we talk about the transfer RNA being like a truck. And the truck, in the back of the truck, you're going to either have things in it or not, right? Trucks are made to carry things. And so at this point, it's full, and it's loaded up with cargo. All right, so I'm going to go to the ribosome now, and we're going to walk through the process of translation using the 3 RNAs, the ribosomal RNA, the messenger RNA, and the transfer RNA. So the ribosome is made of 2 subunits, the large subunit and the small subunit. So normally in our cells, in our cytoplasm, these are actually not together. They're actually detached. And when they come across an mRNA, which is what this is, mRNA molecule, they're going to clamp onto it, and they're going to hold the mRNA in the middle between the large and the small subunit. And we're going to start at the A site, A for amino acid, P for polypeptide, E for exit. So we're going to start here, A for amino acid. Well, which one do I use? We have 64 different tRNAs. Well, I'll match them up. So we have blue, orange, yellow. And so the way I have the matching pairs is blue goes to orange, like the Broncos, and purple goes to yellow, like the Vikings. Easy to remember for this class. So I need to find orange, blue, purple. Orange, blue, purple. There it is. Look at that. It happens to be the tRNA card that holds methionine. Methionine is almost always the first amino acid in a polypeptide protein. This is the initiation phase of translation. It's the start. This is going to shift over one to the P site. Now we have to fill in the A site with the next tRNA card. Yellow, yellow, purple. I'm going to match that up with purple, purple, yellow. Cars parked in garages, apples under trees for RNA. Met, gly. So this is basically the end of the beginning. This is the end of initiation of translation. This is going to move over one. Now we're kind of in the elongation phase of translation. Met actually is going to connect with gly, okay, through a peptide bond. It's a strong covalent bond between the amino and carboxyl groups of the amino acid. That's a dehydration synthesis reaction we learned about a long time ago, it seems like. This card actually is empty now, right? It's not going to get thrown in the garbage, though. It's going to get reused, right? tRNA is going to get recharged out in the cytoplasm. And then we need to bring in our next card in the A site. Purple, purple, blue. I need yellow, yellow, orange. There it is. Proline. So this is the elongation phase. This is the part where the ribosome is zipping along the mRNA, and the sequence, the codes, the codons are getting read by the tRNA, and the tRNA are bringing in the correct amino acid. So elongation phase continues. This thing keeps growing. That kicks out. That shifts over. That shifts over. That connects. That kicks out. We need our last one in here. Orange, yellow, blue. I need blue, purple, orange. Right here. Look at that. It's stop. This is termination. Termination is when we get to the end of the mRNA and it reaches a stop codon. ACU is the anticodon for the stop codon UGA. Okay, if you don't believe me, look up at the chart. UGA, UGA, stop. Stop, right? So what this does, it actually is a molecule that fits into here and blows everything up, okay? It knocks this all apart. It kicks the mRNA out, it breaks the polypeptide chain out, and this just blows up the ribosome too, knocks everything apart. Now the ribosome is going to get reused, right? It just knocks the pieces of the ribosome apart. So the end result, we started with mRNA. That was transcription. We ended with a polypeptide of amino acids. Now, this is not, I mean, in our cells, we're going to have more than 3 amino acids that make up a polypeptide. We're going to have hundreds or thousands, but this is just a little activity, right? This is the protein. This is the nucleic acid sequence. The nucleic acid sequence is the information. The protein is the enzyme or building block of our bodies, right? This is what our body needs as the functional unit to keep us alive. This is the code. This is the information. We went from one biological macromolecule called a nucleic acid to a protein. All right, so that's transcription and translation. I hope that video helped. And if you're, reading the book, pages 200, 201, 202, 203 are the pages that we cover in this topic as well.